Thank you very much. Thank you for the invitation. Thank you for the organizers. Yeah, even if it's virtually, please, as Massimo said, please interrupt. So I prepared quite a long material, but please interrupt as soon as I'm not clear. So indeed, I'll try to share with you some work we've done here in Grenoble. So I'm a physicist. I'm an experimentalist, so I'm not a theoretician, but I'm working with theoretician colleagues, and I will try to share with you what we've done around active suspensions, and in particular how experimentally I use some plankton as a model active particle. So I thought I could just give some generalities about active matter, and probably you might all know what is active matter now, but I will just take some properties I listed here. So it's a matter that is able to consume energy to produce work. So it's an out of equilibrium system by essence. It's composed of the self-propelled units, and the drive is done on each particle, not at the boundary, as we might know from familiar non-equilibrium system. And very often there's an emergence of collective behavior in those systems, as flocking, swarming systems. So with this very broad definition, we can think of many systems ranging from meter scale as crowds to sub-micrometer scale as molecular motor in the cells. So as soon as self-propelled units are composed this kind of new kind of matter. And as physicists, the goal is usually to use the tools we have, in particular in condensed matter physics, to describe and classify the general behavior of the system and try to get to a kind of universal description of the system, even though they can be so diverse, as exemplified here. So in this context, we can define what is an active particle. So it would be the self-propelled unit that compose this matter. And usually it's able of transforming chemical energy into a motion, or at least mechanical forces, with some cyclic transformation. So an example, the best example, let's say, in the living world is a bacteria such as E. coli that is a very model system. And indeed, in those systems, you can observe the emergence of collective effects, and in particular in the bacteria system, if I stay with this one, we can talk about bacterial turbulence. So it's not turbulence in the fluid mechanics sense because it's a low Reynolds number system, but we can observe, as exemplified here, some turbulence patterns in those systems only due to the activity, to the motility of the bacteria. So there are many examples of active matter in the living world, so ranging from fish schools to, again, molecular motor. And I'll be talking about plankton, what you can see here in the ocean. And in all those systems, very often we can observe collective motion and pattern formation that is probably due to the coupling of motility with any other flow or no flow. The counterpart in the synthetic world, let's say, this is an example of a synthetic particle, a Janus particle that has two sides and that has been built to be able to propel itself in liquid, or let's say more macroscopically, those bots that are used to also model active matter, let's say at the centimeter scale. And in those systems, again, some collective motion can be observed, some patterning in colloids here, in quink rollers on the left, or with those bots, you can see a flocking of those robots. So what's intriguing, I think, for physicists is this collective behavior of active matter. It is the dynamically self-assembled system. And the questions usually turns around the onset of this coordinated motion at large scales, how to explain that we can have the emergence of this coordinated motion. And can we talk about non-equilibrium phase transition in this system? So those are the general questions usually in this field. And again, we can be interested in looking at whether there's underlying universal properties on those systems. Yeah, so this is the same information. Here, it's just some examples of what is done in the literature. You can go to more and more sophisticated models for those systems. I think the simplest one, I call it dry model because there's no hydrodynamic involved in this kind of system, is to go from what we know from statistical physics, a Brownian particle that it's made active through velocity. So I think it's the simplest model to describe an active particle. And however, we can already have interacting active Brownian particles. So in this model, we can go to motility-induced phase separation and see a collective motion emerging from the simplest models here. If you consider alignment rules between the active particles, then we can go to Vic-Check model. I'll try to say a word. And then if you want to add the long-range interaction between the particles, you might want to introduce the role of the solvent in those models. So indeed, probably the first inspiring question for physicists was developed by Vic-Check, observing bird swarms the sky. And as a physicist, the proposal was to describe those birds as an elongated particle that has a given velocity and a given orientation. So this is the minimalist model we can think of to describe a bird, let's say. And this particle will be given a rule. Its orientation would be given by the mean orientation of the first neighbors of this particle. And the orientation will be calculated with a little bit of noise added. And with this very simple system, Vic-Check obtained a high density for a high density of particle and a noise, small noise, a polar ordered phase. So this is what I called here the physicist swarm. The observation is made toward a phase transition, toward the dense and dynamic collective phase. This is sorry, I just removed this thing. A phase transition toward the dense and dynamic state of active particles. So when we look at this very simple model of alignment, again, we can imagine this kind of model of active particle would be universal and valid for many different systems. In particular, we can wonder if microswimmers can be a class, let's say, if we can, talking about universality, a class of active particles. So you know of different microswimmers, sperm cells or bacteria are very famous ones, let's say, and but some plankton, some algae are also able to propel themselves. So it's the route I took from the experimental point of view. So I use an algae, Chlamydomonas reinhardtii, and I try to use it as a model active particle. So the reason I chose this cell is several items here. The shape is quasi-spherical. So for the modeling, we can model this as a spherical particle. The exploration, the motility of this algae is really 3D. If we compare to bacteria, for instance, that likes to swim close to walls, this one is really exploring the 3D space. The size is big enough so that the thermal fluctuations can be neglected. So it's 10 micrometer size diameter. And it has two flagella on the top that pull on the liquid to swim forward. And it's quite easy to grow in the lab, even when we are physicists. So this is low Reynolds swimming for this micro swimmer. And what I'm trying to do with this is trying to establish general results that concern active particle, even though I'm using a specific system. And in particular, when relevant, try to look at the role of hydrodynamics in this system. So if you look at under the microscope, you have the feeling here that you're looking at particles. You don't know really that those are living systems. And you can follow those particles swimming around. Here, Pauline and collaborators show a high magnification zoom on the algae, and you can see that this system is using the flagella to propel itself forward. So what I will try to go through, if I have time, is a little bit of details on the complex random walk of this system, just the microscopic description of the swimming behavior. Then I'll show you two examples on how the swimming couples to first a flow, here in this, what I call the photofocusing experiment, and how the swimming is coupled to a complex environment. Here are some pillars, what we can deduce from this. So this is the idea of having general properties of this active matter with a more and more complex environment in a simple flow pattern environment. And if I have time, I'll go to recent results on the dynamic of a crowd of micro swimmers that I will show you. So first, the complex random walk of Chlamyd. So this was the work of PhD students Michael Garcia and Mathieu Martin. If I go directly to the result, please ask if you want experimental details on this, but we follow with standard particle tracking methods the trajectories of the system. What we can measure is that the mean square displacement as a function of time has a two regime behavior. You can see a linear behavior at long time scale, which correspond to a diffusive-like behavior, a random-like behavior. And at a shorter time scale, this can be shown to be a quadratic behavior, where the system is purely ballistic. So if we caricature the trajectory, you can imagine that the system is having a persistent length of swimming in one direction, and losing a loss of orientation at a given rate that is a characteristic time of the system. So in the case of bacteria, for instance, this would be a run and tumble description of the trajectory, or we can look at this as an active Brownian particle too. So here the mean square displacement is just written to connect a linear behavior at long time scale and a quadratic behavior at short time scale. And we can use a connecting law for this. And this will define an equivalent diffusion coefficient that will just define the randomness of the trajectories at long time scale, which is connected to the persistence length we can define and the crossover time between the two regimes, and the velocity that is defined at short time scale for this system. This can be modeled as an active Brownian particle, as I said. So the idea is to consider a particle that has rotational and translational diffusion as a Brownian particle, but the only difference is that a velocity is added, constant velocity is added to the particle in the model. So we use a constant velocity and orientation that is white noise in this system. And with this kind of model, very simple active Brownian particle model, the mean square displacement as function of time can be shown to be diffusive like if the velocity is 0. And then when you increase more and more here the velocity.
Excuse me, Salima,
yes?
A quick question. So in this case, the solvent is without flow. I mean, is just
this is
okay.
dry.
Okay.
Yeah, because you consider constant velocity, you don't consider the interaction. Of course, as soon as you introduce flow and hydrodynamic, the velocity should be modified by the, for instance, the presence of other particles or environments. So this is the simplest model you can. So it's the reason why I call it a dry model.
Okay, thank you.
Then when you increase the velocity, you can see the ballistic regime appearing here at short time scale that is more and more faster. If I go back to the experimental system, this chlamydomonas has another properties. It's sensitive to light, so it's what's called phototaxis. So when because it has an eye spot on the. Can you see my mouse or usually?
Yes, we see.
Okay. There's an eye spot on the side of the cell. So this is supposed to be the simplest eye in the biology world, and it's quite similar to our eye. Actually, it's a model system for research on vision. With this eye spot, it's a light sensor. Let's say when light is detected on a side, a biochemical cascade occurs in the cell that results in a stiffness, let's say, on the flagella that is the cis flagella that is close to the eye. This one will still rotate, and this results in the orientation toward the light of the cell. So this is phototaxis. You can characterize this by measuring the microscopic trajectories of the cells. So when there's no light, as I said before, the trajectories are completely random-like at long time scales. If you measure, for instance, the x component of the velocity in average, you will find 0 for all time because this is isotropic. And now if you shine some light, you can see already that the trajectories are more aligned. And you can measure a typical time response of 2 seconds when you shine light at t equals 0. Then the x component of the velocity takes 2 seconds to take a finite value in average. And what we can define also here is the noise that you can see, that the alignment here, it's not perfect. So it's what you see in the gray region here. If you measure the distribution of orientation, you'll find that in absence of light, those are the black dots, the system is isotropic. When there's a light shining on the system, you can see a distribution of orientation peaked around the direction of light, even though you can characterize a width of of the distribution that can be introduced in the models if needed. So here I showed you rapidly that we can characterize the persistent random walk of this system by defining a typical velocity, a ballistic velocity at short time scale. So this is about 10 times the diameter per second. A reorientation time of the system that is on the order of seconds, and then diffusive line behavior at long time scales. And in fact, those characteristics are very generic behavior for microswimmers. You can find the same dynamics, even though for bacteria it's more abrupt change of orientation. The microscopic dynamic can be recovered in bacteria or in synthetic microswimmers in the model of active Brownian particles too, or even crawling cells. So we can use this experimental system to be quite generic for many systems. And also the Phototaxis response that characterize the chlamydomonas, the synthetic photosynthetic algae, can be well characterized and quantified, and can represent what are called the taxis in the general sense. Some systems are sensitive to chemical gradients or to gas gradients. So we can use phototaxis as an example or representation of a generic taxis. So don't hesitate if you have questions on this general part already.
Yeah, I have a question,
yes.
Salima. Sorry, when you have shown the PDF of the orientation, so the slide before this one, the PDF, okay, this one. So light is, I mean, the way you depicted, the light is on the horizontal axis. So, right? Because it's Vx.
Here, no, yeah,
So while at the same time there is gravity, I mean the gravitaxis in the algae, right?
Yeah.
So I would have expected I mean some non-symmetric distribution of the orientation, just because they will without light they will tend to go up. With light they want to go horizontal, so there should be a balance. While it seems that the distribution is completely symmetric. Yeah.
So to to answer for this, first here this strain is not so gravitatic, so it's not
okay. It's a low gravitatic strain. Okay, thank you.
But still some gravitatic strains are used also. And here what I call horizontal is the light is here and gravity is perpendicular. Mm-hmm. So as we use 2D projection of the trajectories, we wouldn't see the effect you're talking about. It's not in the same plane. Yes.
Okay, thank you.
Thank you for the question. So now if we go a little bit beyond and with just coupling this system with a very simple flow, I will try to show you what we call the photofocusing, so coupling with the flow and phototaxis. So this was the work of Mathieu Martin during his PhD and Chabel Garcia during a master thesis a few years ago. So what you see here, it's a suspension of active particles, so suspension of chlamyd, flowing, made to flow in a pipe, so with a very simple Poiseuille flow. And what you see at a given moment, the cells are all concentrated in the center of the channel. So what happened in this experiment is that we use a simple Poiseuille flow together with a light stimulus on upstream compared to the flow. So the particles are made to flow much faster than the velocity of swimming. Okay, so there's a factor of 20 or 30, I think, between the velocity that we impose and their swimming velocity. But still, there's a coupling that appears when light is on where the cells are focusing in the center of the channel. So this is reminiscent of what Kessler has observed before with another taxis, let's say gravitotaxis, as mentioned just before, where cells were observed to focus on the center of a pipe because of the coupling with gravity. Gravitotaxis, so this ability of cells to swim against the gravity. So it's the same behavior. And so this kind of system can show that even in turbulence, when the velocity of flows are much faster than the swimming velocity of some plankton, it can be deduced that the coupling with motility can still be relevant in turbulence flow to explain patchiness that can be observed in turbulence. So in this experiment, in our experiment here, the mechanism is very simple. So if you consider a particle that is sitting, let's say, in the bottom half of the pipe, so this particle has a preferent orientation toward the light, so here to the left, let's say. And meanwhile, it's subjected to the vorticity of the flow because there's a shear, local shear around the particle here because of the Poiseuille flow. So you have a higher velocity on top and a lower one. So the particle is subjected to vorticity. And those two ingredients together lead to a net velocity across the flow. So because of the swimming velocity of the particle. So even though the translational flow velocity is much larger than the swimming velocity, you can see a coupling between because the swimming velocity will lead to a migration toward the center of the flow. Of course, this argument can be inverted. If the light was on the right, then the particle would go toward the wall. This you can describe statistically. So it's what we've done with in collaboration with Eric Bertin in the lab.
If I interrupt you, Salima, yes? I see a question on the chat by
sorry,
Raquel.
Okay.
So I can read it. A recent PRL paper by Cortese and Won showed the clammy rotates as it swims and undergoes helical swimming. They suggested an alternative mechanism for Phototaxis involving modulating the sign of biflagellar dominance. Have you seen this behavior? Could differences be due to different strains?
Oh, thank you for this reference. No, as I showed you, the manner we look at the Phototaxis is a simple microscope, let's say ensemble description that we do on the trajectory. So I'm not sure I could detect this kind of effect, but I will have a look to this. Yes, thank you. So please, yeah, don't hesitate using even your microphone because I'm not sure I can look at the chat. Yeah, as I said, so you can try to describe the dynamics of the swimmers during the coupled with the flow as a statistical description of the system. So if you look at the probability for a swimmer to be at a position, to given position R at given direction and time t, what you can write is a simple, so you can consider a point-like particle even here that has a given swimming velocity that is constant. So again, we don't take into account here probably the hydrodynamic feedback on the particles. So there are point-like particles with constant velocity of swimming subjected to a parabolic flow. So they're transported by this parabolic flow. They have a constant swimming, and they are subjected to the vorticity of orientation. So we can consider that the orientation of the particle is just rotated through the vorticity of the flow with the with the constant here parameter that can be adjustable. That reflects a hypothesis we had on the rheology measurement that I'm not showing here. That particle can resist a bit the vorticity in those systems. And on the other side, you can take into account the random reorientation of the cells. So they have a rate of reorientation that we assume is still the same here, even though there's a flow. And here, what we take into account is the noise in the orientation distribution, as I showed you before. When light is on, all the cells are not exactly oriented toward the light, but we take into account the distribution, the experimental distribution we measured with the given width that is experimentally defined and not adjusted. So with this, I will not enter the details of the result, but just to show you that with this simple modeling, we can quantify quite successfully the experiment. We can measure the width of the focusing length, the way, the time or the length it takes to be constant, and the distribution of cells in the channel. So all this is in good agreement and has no adjustable parameter except for this beta parameter that I added in front of the vorticity, and this backs up a hypothesis we made before for rheology that the cells are able to resist a bit the vorticity. So the take-home message here is if you look at the ingredients we used to model this behavior, you can observe that this has no specificity to the cellular system. Parabolic flow coupled with a given constant velocity subjected to reorientation, and that has a random reorientation and distribution of light. So in the model, there's no specific characteristic related to the biological system that we are using. So you can deduce that this is quite a generic result on the coupling between the flow and the particle. And in particular, there are some questions related to immune systems when some immune cell or motile cells of the immune systems are transported by blood flow. The motility has always been taken as irrelevant because the blood flow is too strong, but probably this kind of modeling can show that sometimes the motility can be relevant even though the blood flow is important. What time is it? I hesitate. Maybe I go through this part quickly because I would like to have time to go to the last one. So if I'm not clear at all, please interrupt. But I will try to show you quickly how active particles behaves in a complex medium. So this was the PhD work of Marvin Brun-Cosmo-Bruni. So the idea here is to try to look again with the same approach, which is ensemble average of many trajectories. We look at the particles inside the complex medium formed here. It's a microfabricated medium of pillars, cylindrical pillars, where we can vary the distance between the pillars. And then we can track all the particles again in a 2D projection. We don't have 3D tracking, but it's not necessary here for what I will present. The first observation we made is that the orientation of the of the trajectories are not isotropic. In particular, what we can see is there's a more abundant trajectories in the directions of the alleys between the pillars than at 45 degrees here. So the trajectories are less represented in this direction than those direction here. We can see here some particles that are traveling here completely straightly in between the pillars and less in the 45 direction. So inspired by this first observation, we modified a bit the previous model. So this is still an active Brownian particle. The velocity has a constant velocity. So there's no coupling with the environment, no explicit hydrodynamic coupling. So this is a simple active Brownian particle. And the medium has been included in the model as an effective medium. So instead of trying to model the pillars, what has been added in the model here is that the frequency of reorientation will have this shape, let's say. So we just increase the frequency of reorientation, the probability of reorientation in the direction corresponding to the 45 degrees, the diagonal direction in the pillars. So this is the way we add the effect of this effective medium, let's say, on the dynamics. So we just add an anisotropic reorientation frequency of the active particle. And with this, we can properly fit the angular distribution that are found experimentally. So those are the lines, are the fits that are deduced from this model. And from each fit, we can deduce a value of the isotropic factor that we use in the frequency reorientation. Okay. And with this value fitted for each experiment, we can deduce analytically this broken line, the effective coefficient diffusivity of the system as a function of confinement that is related to the distance between the pillars, and compare this to the experiment, and the experimental results are very well described by this simple model. When we change the confinement, when we increase the confinement, so decrease the distance between the pillars, the diffusivity of the cells is decreasing in this way. So here we proposed a very simple dry description of the microswimmers. Again, this was enough to describe the experimental results quite successfully with an effective medium approach, but still we use a symmetry in the frequency anisotropy reorientation that is hidden in the choice of parameters. So we chose only one parameter for this frequency anisotropy factor, and the question we rise here is are hydrodynamics hidden in this hypothesis? So how to explain the channeling of the cells in between the pillars? Is this due to hydrodynamic, for instance? So we propose here a dry model, but probably behind the choice of the hypothesis here of the parameters, probably some hydrodynamics can be hidden here. Okay, so I'll go to ongoing work here of Eduardo Alalam, a PhD student. So we use our favorite system, the model system, to explore the dynamics of a crowd. So you probably heard about crowd dynamics. That is a quite fascinating topic nowadays, what you see in this experiment is some people that are asked to evacuate a room. On the left movie, they are asked to go gently, slowly, let's say, with a small velocity to evacuate the room. On the right, they are asked to go as fast as possible. So you can see that there's some fighting at the door here. And the conclusion of this is what is called faster is slower effect. The people who are trying to get as fast as possible outside are evacuating slowly the room, whereas the other had evacuated already the room. So this kind of paradoxical effect in crowd motion has intrigued quite a community of physicists in particular. And there's a very nice proposal of Zurigel and collaborators, where they in this paper they propose to quantify many different systems, suspensions, grains, or even ships, with the same phenomenological approach and the same observable in those systems. So the question is, again, as a little bit what I presented for active matter, is there any universality that we can recover in those systems, just like clogging effect, as we know for sand, in those crowd motion? Here, the illustration is another example of what is done in crowd motion. If you put an obstacle just in front of a door, you can, with some adjustment, fluidify the evacuation of the crowd, depending on how you place this obstacle. So the question we ask is, a slide is missing probably. The question we ask here is, does a wet system, a system of active particles immersed in a fluid with long-range interactions, typically because hydrodynamics is intrinsically long-range, can we fall in the same phenomenology of crowd motion, or will we observe a new behavior or a new class of system because of hydrodynamics? Maybe I can say already that the phenomenology is found to be very similar, and we didn't manage to extract a typical specificity of the hydrodynamic system. Anyway, what we design here is a cell, like an hourglass shape construction. The system is 2D, because crowd motion is usually observed in 2D. We are not in 3D. So the height of the channel here is 20 microns, which is about 2 times the cell diameter. And we can vary the size of the door here. We use light as a trigger of motion in this system, so there's no flow. The cells are gently swimming through the constriction because they're swimming against the light stimulus. This is a strain that is negatively phototaxis. And what you see is the building up of a kind of clog here before the constriction, and the cells are still going through the constriction one by one, even though it's very dense before. What we can measure, so the first case here is for a large door, so there's no problem. Cells can swim through the gate. So what you can measure here is the cumulative number of escapees, so after the constriction, as function of time. And we vary the velocity, the swimming velocity of the cells. So this is done by adding some polymer in the solvent, in the surrounding fluid, to slow down the velocity of swimming. So we can vary a bit in this range. So somebody is asking to Yes.
Yes, there is a question by Klaus. Klaus, you can unmute.
Yes, sorry for interrupting. I wanted to give you the chance to finish your sentence, actually. But the question is, did you say whether the light reaches all the cells or only part of them now? Because that's not so obvious from your picture.
You mean some shadowing effect that you could expect?
Something like that. Yeah.
Apparently, experimentally, it's difficult to put a sensor there. But those cells after the construction, they're still ballistic. They're all ballistic. So apparently, their response to light, even though it's a cloud. Yeah. So
they all see the same
varied
light, basically. That's the answer.
Yeah, the velocity. We measure the velocity distribution on the left side of the cell, the geometry, and we measure the velocity distribution and the exit. Even here we tried to see if there was a fluctuation in the velocity modulus. We didn't measure something significant. So the distribution of the population in terms of velocity, for instance, is similar before and after the construction. Yeah, I was expecting probably an effect on the velocity, but we couldn't see a significant effect just at the exit. So yeah, the response probably also because it's a real 2D system. I'm not sure. So we vary the velocity of swimming. We can decrease the velocity of swimming. So this train is swimming fast, 120 microns per second. When I give the velocity, there are always the mean velocity measured on the distribution of velocity in the experiment. So sometimes it varies from a curve to another one, probably, because we measure it on site each time. And we can decrease it, you see here, a factor of 3, let's say, by adding a viscous component in the surrounding fluid. So this way we can explore the effect of velocity. As I showed you, the faster, slower effect, for instance, for crowd motion, we can try to explore it here by decreasing the velocity. So here it's nothing exceptional. If you measure the cumulative number of escapees after the construction as function of time, the more you wait, the more cells are going through. And actually, this is you can rescale this by their velocity and their size directly. You can see that the cumulative number of escapes are well described by boring law. Another observable that has been proposed by Zurigel is what is called the survival function. So this is the cumulative probability distribution of time lapses. So you can look at the time difference between two successive exits at the door, and you measure the cumulative probability of this distribution, again by varying the velocity and the shape of this survival function. In this case, because the door is large, this probability is exponential, as you see in Lagrangian plot, and the same rescaling time is used here for the different curve, different velocity. So this is, let's say, the boring case. There's nothing that prevents the cells to go through this large door at every velocity. And when you go to a very small door, 10 micrometer, which is about the cell diameter, you can do the same measurement. And already here you see a strange behavior, because this pink one, it's too pink, I don't know. This curve is the slowest cells. So they're swimming at 48 microns per second in average here, and still they're exiting the construction more often than the faster ones. So you can see an inversion here. The velocity, the the velocity increase, the flux is decreasing. And here what happens, this is to show that the rescaling doesn't hold at all here. It's obvious. No, it seems that it's the same. No, yeah, still. Anyway, you can see that you cannot rescale because the direction is the opposite. And when you look now at the survival function, what you can see when the velocity is modified here. Here the representation is a log-lin scale. So you can see that the exponential is not recovered here, and what you see on the log-log representation is that there's a power law behavior that emerges at long time scale. So what does it mean? It means that you don't have any more a characteristic time as an exponential would suggest. You cannot define an experimental time of exit. And what you have is that longer time lapses between the exits are more and more probable, let's say, more and more can happen more often. So this is the emergence of the power law behavior at long time scales, that longer and longer times can appear at the exit at the construction between two exits of cells. So this is what we can call here a jamming regime, let's say clogging in the crowd motion terminology. And that is described here by a power law decay, or let's say the birth of a power law behavior at long time scales. And what is shown here as function of the velocity might resemble a faster is slower effect. So in the sense that there's a increase of the probability of finding larger time lapses when particles are faster. So this is paradoxical. So what we are into now is we produce the active jamming. Maybe I can do a survey here among you. Do you like active jamming, or it's not the right terminology for this? Because I found that it's not exactly clogging as we have in, let's say, in granular matter. When there's clogging, I think it's not flowing anymore. I prefer this word jamming, but I'm not sure the community would accept. Active jamming phase diagram here. The size of the construction is plotted as function of the velocity. And for instance, when so evidently, when you decrease the size of the construction, you go from a flowing regime to a jamming regime, and there's a boundary. And when you increase the velocity of the swimmers, the jamming regime occurs for smaller doors. So I was myself quite surprised that we could get to such very dense active suspensions. I thought hydrodynamic interaction will prevent to go to random maximal random packing is what we measured just before the constructions. We see some clogging events, if you allowed me to call them like this. So there's longer and longer time lapses between the evacuations, and we observed a paradoxical evacuation dynamics that we think may resemble faster is slower, even though I'm not sure it's exactly the same origin in these systems. And in the perspective, as to go on with the analogy with crowd motion, we'll look at the effect of obstacle on this jamming in this system. So with this, I would like to thank you again for organizing this workshop. And I will mention again the different PhD students that worked with me. Actually, Eduardo is missing on this list. I don't know why. It should be there. Oh, okay, Eduardo is on the right. Eduardo LLM is working now on the crowd motion, and all this work is done usually in collaboration with the theoreticians, Philippe Pella in my group, Eric Bertin in the lab also, and Walter Zimmermann in Bayreuth, or Bertrand Maury, who is a mathematician working on crowd motion models in mathematics. We're discussing the crowd dynamics with him. So thank you very much for your attention. I hope this was more or less clear.
Thanks a lot, Salima. It was very nice. Especially the last part was very interesting.
Yeah, thank
you. So it's time for questions. We have some time for a few questions. Please either unmute or write on the chat. Raise your hand if you like to ask a question. So maybe I will ask a question. Okay, no, there is a question by Klaus. So I give the Klaus. You can ask.
Yes, thank you. Thank you for the nice talk. What you called active jamming, it looks like a very special situation because the light direction from one side matters for it, right? It is important where the light source is for this phenomenon. And so also in jamming, if you add activity, you usually can also melt the jammed state. So my feeling is that that active clogging would be a more appropriate notion for exactly this particular experiment you described.
But can't you say that clogging would be also the same way removed by activity?
Could be, yes, but in your case, it's really special, right? You have flow and you have the activity, but the activity is in a particular...
No, there's no flow. There's no flow. There's only activity.
So it's only the activity, but it is still in a very special direction, right?
But you can forget of light. It's some active particles that have a preferred orientation. So if you go to the model, you have an active Brownian particles that have a preferred orientation. So the orientation are not isotropic, but just preferentially oriented given the distribution I gave. So those would be the minimal ingredients. And yeah, probably it's equivalent. I don't know. I had in mind that jamming retained the notion of flowing, contrary to clogging. When we talk about jamming, but
is the direction of the light, it is important, right? Because the particle have a particular reaction to the direction of the light, not only to the fact that there is light, but it matters from where it comes.
Yes. Yeah. No, but what I mean is that at the end, the light is used just to bias their swimming behavior. So if you go to the minimal ingredients you need, it's just an active particle that has a velocity, but this velocity is not isotropic, but has a distribution of orientation that is picked around one direction, that is the horizontal one, let's say.
Which is a bit like a superimposed active flow, so to say. Well.
Yeah, it's different. Yeah, because there's because it's locally driven. So so if there's a reorientation because of interaction with the neighbors, for instance, each cell is able to recover its own direction, which probably wouldn't be the same coupling with the flow. Yeah.
Thank you.
Other comments, questions? Maybe I have one. So coming back to my previous question concerning phototaxis and gyrotaxis, gravitaxis, you say that there was no gravitaxis there. So I was wondering if there is some experiments you did or other groups did in which you have both phototaxis and gyrotaxis. I mean, maybe in different directions or stuff like that. If some phenomenon emerge, or you think something interesting would emerge.
Yeah, I tried to do some experiments on that that way. The type scales are very different actually. So at the observation scale, I think probably because I did microscopy observation. So at the scale of the microscope field of view, I think phototaxis is a fast response, and gravitotaxis is it's slower. You see, it's you need a larger time scales or larger length scale to observe the effect of gravitotaxis on a population. So I didn't manage to really couple those bias, but I think there are you see in all the bioconvection experiments, or the literature in bioconvection. I think in some papers they are coupled without being explicitly mentioned, but just because of the observation, they can couple in. But I think because
in terms of time scales, you say that phototaxis is faster. So can you quantify the time scale?
So for phototaxis is what I showed, like on the order of 1 second, the whole population is oriented. For gravitotaxis, I think in Kessler, the effect is I'm not sure. So this is intuitive answer, but I haven't measured the response time.
Okay, but this is speaking about the population response time, not a single cell response time.
Yes.
Okay.
Yeah, and the only thing is that because I've started with phototaxis measurement, and probably to couple the two, it would be better to observe the cells at the bioconvection scales, let's say, which is usually millimetric, centimetric, to observe those patterns, and to add light on top of this. The other way, for instance, in those small cells, in those small setup where I'm observing 200 micrometer range of trajectories, I wouldn't see a bias because of gravity. For instance, in a thin channel of 200 micrometers, you don't see all the cells on top of the channel. For instance, they are evenly distributed. If you follow them really over a long time, probably they would be on top eventually, but it's not some gradient that you can see at those scales.
Okay, thank you.